Ladies and gentlemen, good afternoon. Antibiotic resistance, how serious can it become? Well, let's start with the situation in Europe these days. These days, about 25,000 persons die from infectious diseases every year. Last year, most of you have read the O'Neill report, giving an analysis. How is the picture in 2050? Well, look at the situation in 2050. In Europe, about 8 times that number. Look at Asia. It's almost approximately the Norwegian population will die every year from infectious diseases if we don't find new cures, new ways to deal with the problem. This is not science fiction, but we need life science. ABR, how did it start? Well, before the invention of penicillin G, Alexander Fleming in 1928, it was a dangerous time. You could die from a wound in your index finger. Having a lung infection was very dangerous. About 1/3 of the patients died. Giving birth to children was dangerous. So, in 1944, Alexander Fleming was on the front page of Time magazine, stating that if we don't use these drugs correctly, we will see resistance in the future. That's amazing. Was he right? Indeed. Look at this. This is a time scale. Above the time scale, you can see when the respective great drugs were introduced on the market. Below the scale, you can see with the same color code, when did the world see resistance? Look at one of these examples with cyan-methicillin, introduced on the market in 1960. 2 years after, the world for the first time saw MRSA, methicillin-resistant Staphylococcus aureus. There's another thing with this figure. Have you looked at the amazing void in new projects? You could call it an industrial silence. Why? Well, imagine if it costs, let's say, $1 billion to develop a drug. Would you have paid for such a project if the drug doesn't work after 2 years? They don't get their money back. So the result is only 3 genuinely new classes of drugs in the last 30 years. That's 1 drug per decade. So, what's the reason? What caused this? Well, we caused this. It's our reckless use of antibiotics. It's how we use antibiotics in agriculture. We use antibiotics prophylactic to breed animals. It's a lack of regulatory control in large areas in the world. And lack of prescriptions, lack of new projects. So this star is a good illustration of what caused antibiotic resistance. I was so lucky to be invited to India, together with other researchers in Norway, to start collaboration on antibiotic resistance together with Indian researchers. And in our New Delhi trip, we went into the back streets. I was frightened. There you can find shops where you can buy all the modern antibiotics and cancer drugs without prescription, along with your Coke and sausage. No wonder why we have this problem. So, now I will tell you the story about what we did. If you look at the situation today, let's pick one of the carbapenems, meropenem. We are so lucky to collaborate with the KRES lab in Tromsø, Ørjan Samuelsen. There they have all the worst bugs. We are so lucky to be able to test on the ESBLs, gram-negatives. Meropenem, no effect on these bugs. Our concept, our lead candidate, could you guess what's the result? No effect. But when we bring them together, an amazing thing happens. Resistance is gone. Meropenem works again. So what this slide tells us, our drugs are not new antibiotics, they are adjuvants. They actually remove resistance in the bacteria, so meropenem can work again. Well, this must be very dangerous for us then. Very tough drugs. ZinChel is founded on binding zinc selectively in the bacteria. So that must be dangerous. Well, we have done studies now in live mice. There are no clinical signs in large doses, in vitro and in vivo. So what about red blood cells? Red blood cells contain iron. Well, a zinc chelator must be very dangerous. We have done this study, and in large doses, nothing happens to our red blood cells. Now we are in the middle of doing infection studies in live animals. That's a very important study that we do in SSI in Copenhagen. And we also need to study what happens with resistance with this concept. Is it the same thing? You remember the time scale? Is it so that our concept also will see resistance in 2 years? Well, we need to find out. That's possible to study in the lab during a short time. So, now a message. How do we deal with this? Well, I'm very happy to see that, at least also during this conference, that political forces and global health authorities now engage. But it's very important that global health authorities, together with political forces, internationally and nationally, join the forces. And two important things must happen. We must stop the reckless use of antibiotics globally. The second is that we need rich funding. Research and innovation is necessary to deal with this, because the pharmaceutical industry is the only institution that have the competence to develop new drugs. So since they don't want to do this because of lack of income, we need to support them so they can develop new drugs. Without this mechanism, I think I'm pessimistic. So, to sum up, 30 years and 3 generally new classes of drugs. And none of them work against the worst bacteria. So now people are dying. No cure. The ZinChel project is a hope, but now not without conditions. And it's a long run to develop a drug, you know that. So we will try. Depending on the results, we will start this year, hopefully starting a company, and try to get this concept out to the patients as soon as possible. Well, I'd like to thank my fantastic project group and all my collaborators that has made this possible, and the places that we do this work. And not the least, without this funding, it would have been impossible to do. And especially the Norwegian Research Council and the Biotech 2021 mechanism is a fantastic mechanism. Not the least, Novo Nordic. They have a mechanism that we don't see in Norway. Novo Pre-Seed actually gives funding to projects, good projects, no strings attached. Easy money. Pre-Seed is one very important mechanism that still is a bit slow. So I really hope that at this point we can do something. So with that, I thank you for your attention.